I'm here to introduce Tony Prestigiacomo because I can say it.
I can say it.
I know you can. Try it off. We both practicing this. Tony is with the Department of Environmental Conservation, the local hub, which he's going to tell us a little bit about. Tony is generally local. He's from, I kept his intro from our annual meeting, so I'm going back to my notes here. I'm going to embarrass him a little bit. Tony's from Herkimer County. He's born and raised there. He went to SUNY ESF in Syracuse. He has a DS in environmental forestry, biology, and an MS in water resources engineering. For 17 years prior, he worked as a scientist at Upstate Freshwater Institute in Syracuse. His main focus at that time was monitoring and assessing lakes and reservoirs and streams, and mainly on the New York City reservoir system and Onondaga Lake. But it also included monitoring of eastern Finger Lakes for various projects. In April of this year, he joined the Finger Lakes Water Hub, focusing on watershed and water quality issues specific to the Finger Lakes region. And we happen to be lucky enough to be one of his lakes here in Canandaigua. And Tony's an expert because he lives far away in Cortland. That's how you know if you're an expert. Far away. With his wife and 2 daughters. So I'm happy to introduce Tony Prestigiacomo. Take it away.
Thanks, Wade. The volume okay? Volume good? Okay. All right, well, I feel very lucky to have you as one of my lakes, so it's very nice to be here again. I'm going to talk to you a little bit today about phosphorus, the different forms, why they're important, and a term called bioavailability. So a quick little outline. I'm going to talk about lakes and water quality in general, phosphorus and why it's important, sources of phosphorus to lakes, eutrophication of lakes due to excess phosphorus, forms, and then a few case studies, including a neighbor of yours to the east, Cayuga Lake, a few other case studies, and then just wrap it up with a little bit of management considerations. So this is my reminder to let you know there will be a lot of acronyms. I will try to pause and spell them out for the first time. But if I don't care if you ask questions during the presentation. So if I forget something, just please let me know, and I'll try to remember to get to it. So limnology is the study of lakes. I like to think of lakes as having genetics. So how they're formed, whether by glaciers or animals or drowned riverbeds or man-made reservoirs, their geology and underlying chemistry, hydrology, what comes into a lake, what leaves a lake, how long the water stays in any particular lake, and its shape, size, depth, area, volume. All of those things are very important lake characteristics, and they all affect the natural underlying water quality of a lake. So as part of the reason that Canandaigua Lake is a deep, beautiful, clean, relatively clean lake, and a reason why a smaller farm pond like this one will never ever get to be Canandaigua Lake. I get asked quite a bit, why does my pond not look like such and such a lake? And it's because it's shallow, it has a lot of nutrients. Your lake, it is what it is, there's nothing wrong with it, but it'll never be a skinny Alice or a Candy. So temperature is very important in lake water quality. The pointer's not working. So the graph over to the right-hand side is a graph showing temperature with depth. Okay, so the top is lake surface across the horizontal axis. Depth is the y-axis, and what you have is temperature with depth. Okay, and a characteristic of freshwater temperate lakes in our region is that they will thermally stratify in the summer, which means the warm upper waters will be separated from the lower waters. We call these warm upper mixed layers the Epimenian or upper lake, and that's where a lot of the primary production, algal growth, things like that happen in a lake. There is a middle layer called the Metalimnian, where temperature changes very rapidly. You can see between the dashed and dotted line a rapid decrease with temperature with depth. And then over top of all of those is the Hypolimnian of the lower water layer. It's cold And interesting is it's isolated from the upper layers. So these lakes, like Canandaigua, will normally stratify in the late spring and early summer. And because of the density of water is very temperature dependent, so there is little or no mixing between these different layers during the summer. There's a lot of mixing within the layer, but there isn't a lot of vertical transport of nutrients and other things up and down through the water column. And a lot of more eutrophic lakes, or high production lakes, the hypolimnia itself is cut off from the atmosphere, and that's why through bacterial respiration, it uses up oxygen. That's why a lot of these lakes that are very eutrophic will lose their oxygen in the bottom waters during the summer. So trophic state. Eutrophic lakes, which Greek means well-nourished. They are highly biologically productive with excessive algal and plant growth. Due to high nutrients, chlorophyll A is kind of the surrogate of algal biomass that we use. They tend to have poor clarity, poor aesthetics, prone to persistent kind of long-term algal blooms, with little or no oxygen in the lower waters during the period of thermal stratification. And some of the lakes are prone to kind of rotten egg smell, hydrogen sulfide odors in the summer also. Mesotrophic lakes are kind of that middle level, so they have moderate levels of algal production. Nutrient levels are usually moderate, usually pretty good clarity. They can have minor aesthetic problems, other problems as well, maybe short periods of low oxygen in the lower waters during very short periods of time in the summer. And they may have occasional algal blooms. Oligotrophic lakes are low nutrient, low biologically productive lakes. They generally have low chlorophyll, high visibility, very minor, no aesthetic problems, and there's an asterisk there, which I'm sure you all are aware of recently with the algal blooms and the finger lakes. But in general, these low production lakes are what we consider pristine lakes, and they have fully oxygenated water columns. And so you may have heard the term eutrophication. Eutrophication, you can think of that as natural lake aging. So lakes are basically sediment and water retention basins, even natural lakes. So as a lake is formed and as it ages, it collects and retains nutrients and sediment over time, and it kind of fills in over time. This is long periods of time. And there's an old adage from lake to meadow. So as a lake kind of is newly formed, it'll gradually lose depth as it gets sedimented in, and eventually that'll turn into a pond and a bog and a meadow, and pretty soon, after centuries and centuries of time, it'll cease to be a lake. And that's natural. Cultural eutrophication is an acceleration of that natural eutrophication as a result of human activities. Land use changes and development, the presence of agriculture, modifying hydrology, leads to excessive sedimentation, nutrient inputs, which result in algal and plant growth, and that cultural eutrophication can happen over much, much shorter periods of time, decades. Watersheds, and here's a nice map of the Canandaigua Lake watershed, is the land area that drains to a water body. And that's very important in determining the water quality of the receiving lake. Things like slope steepness, what the topography is, land use, population density within the watershed are all critical factors. And one of the most critical factors actually is something that we really don't have much control of, and that's the watershed area to lake area ratio. So all that is you're taking the land area and dividing it by the surface area of the lake. And so what you see if you do that, lakes with large watersheds to lake area are generally poorer water quality lakes. And that's due to high nutrient water inputs relative to the lakeside on an annual basis. And on the other side, smaller watershed area lakes generally have better water quality.
And where do we fall in that ratio?
I'm getting right there. No, good question, very good. So this is another graph. On the x-axis here is the watershed area to lake area ratio. That D-less means it's dimensionless. There's no units. And on the y-axis here is summer average chlorophyll. Chlorophyll, again, is our surrogate of kind of algal production, algal biomass. And this data is from the late 90s. I'm sure some of you are aware that one of the tasks of the new hub is to kind of find out what data exists. I know that Bruce is out here religiously collecting lots of good data to analyze. And I hope to update this with his data set and the CSLAP data set this winter. So each dot here represents summer average values of chlorophyll for each of the Finger Lakes in the late 1990s, okay? And what you can see is Skinny Atlas Lake, SK, as the lowest watershed to lake area of all the lakes, okay? Around 4, which is very, very low. And as a result, it has the lowest summer average chlorophyll concentration. Makes sense. And you can see the different ones. Sorry, laser pointer's not working. I circled Canandaigua, that's the CG. You're there kind of in the middle in terms of watershed to lake area ratio, but very, very good water quality. And there are other reasons that I'll get to in a minute. Owasco Lake of the Finger Lakes has the biggest watershed area to lake area ratio, somewhere around 17, I think. Which is fairly large. So Owasco kind of has one lined up against it from the beginning. It has a huge watershed compared to the size of its lake, which presents all sorts of water quality challenges. Honeoye and Canisius are up here, kind of in their own little circle. Not overly high ratios, but a lot of chlorophyll, and there are other reasons that I'll get to that in a few minutes. So one thing Canandaigua has going for it is it's a really big lake. It has a big watershed with not an overly huge surface area, but it has a lot of volume. There's a lot of space for nutrients and sediment to come into the lake and kind of settle out and not impact water quality quite as severely. So this graph on the x-axis now is volume in billion gallons, and then the y-axis is summer average chlorophyll again. So you can see, due to its kind of retention size, its volume, it helps it in terms of its water quality. Okay, so that's my quick little throat clearing on limnology, and we'll get down to some phosphorus. So why is phosphorus important? Phosphorus is a macronutrient. It's required in large quantities by both animals and plants. It's a major cellular component of algae, plants, phytoplankton, enzymes, DNA, respiration, metabolism, cell wall components. And you may have heard that it's phosphorus is often the nutrient that limits or restricts algal or phytoplankton growth in freshwater ecosystems. So the old rule of thought is you cut the phosphorus, you cut back the algae, right? Try to limit the amount of phosphorus that comes into the lake. So why is it limiting? How do we know that it's limiting? Well, in the late 1800s, early 1900s, oceanographer named Redfield, he traveled all over the world's oceans and did nothing but collect phytoplankton and analyze them for nutrients, right? And what he found was in oceanic algae and plankton, there's almost nearly a constant ratio of nitrogen, N, to phosphorus. And that ratio is, on a mass per mass basis, about 7 to 1. So in an algae, there are 7 parts of nitrogen for every 1 part phosphorus. The Finger Lakes usually have nitrogen well in excess of algal needs. So some common N to P ratios, and again, these are from the late 90s. You know, Canandaigua has almost 80 parts nitrogen for every 1 part phosphorus in the water. Cayuga has about 130 parts nitrogen to 1 part phosphorus. Honeoye and Canisius, they're closer to that 7 to 1, but they're still usually in excess of excess nitrogen algal needs. Now that's not true at all times and in all places in these lakes, but generally it's phosphorus that limits algal growth because it is the nitrogen is in such excess. And so here's another graph to kind of hammer that point home. It's the same one circle per lake, so all 11 lakes. Summer average phosphorus on the x-axis, and then summer average chlorophyll on the Y. And what you see is a nice, strong, linear trend of when the phosphorus goes up, the chlorophyll goes up. Okay? So it's phosphorus that's kind of driving the chlorophyll or the algae. And on the other side, so what I've done is I've taken the chlorophyll and put it on the top axis, and then moving down on the y-axis is summer average secudisc, or water clarity, right? So if you envision that the lake surface is here, the deeper the secudisc, the clearer the water, okay? And what you can see when the chlorophyll is low, so close to zero, the clarity is really, really good. And on the other side, when the chlorophyll is on the higher side, the secudiscs are quite a bit lower. Okay. So there are lots of sources of phosphorus to lakes. Atmospheric deposition has some phosphorus. It's usually very small relative to other inputs. There are point sources to lakes, which can be substantial. Those are things like wastewater treatment plants, concentrated animal feeding operations, industries, and point source contributions, especially from wastewater treatment plants and industries, their contribution of phosphorus to lakes has been cut back substantially due to the passage of the Clean Water Act and phosphate detergent bans and things like that. But depending on the lake and the treatment plant, they can still be substantial sources of phosphorus. Non-point sources or diffuse sources from the watershed, they usually dominate in the Finger Lakes. Things like phosphorus runoff from farm fields, developed lands, residential lands, use of fertilizers, leaky septic systems, leaky sewer systems in cities, forests, and string banks and ditches can all contribute phosphorus to lakes. And there can also be internal sources. So these are internal to the lakes themselves. So, and they can be substantial in the case of Honeoye and Canisius, kind of shallower, more eutrophic lakes. When the hypolimnion, we talked about earlier, has no oxygen or low oxygen in the summer, phosphorus can be released from the sediments and kind of accumulate in the lake's lower waters. And in a case like Honeoye, which mixes often, those nutrients are brought up to the surface where the algae can use them. Even in the case of a lake like Cayuga Lake, you will get some internal recycling by zebra mussels, quagga mussels, that can accumulate in the lower waters. And when the lake, they call it, turns over, completely mixes in the fall, those nutrients mix throughout the water column, stay mixed, and then are available next spring for the algae to use when it's warm. Okay, so there have been numerous studies that have documented degraded lake water quality or eutrophication due to excessive phosphorus inputs. Here's a list of some of them. I'm not going to go through each of them, but the common theme is that if you limit the phosphorus loading or the phosphorus input to lakes, you reduce the primary production and you improve the water quality. These folks, these are kind of some of the more classic studies. And also that phosphorus loading is critical in relation to the size and shape of your lake, which we've talked about a little bit, but I'll go through that a little bit more detail in just a minute. So one of the more famous studies was by a researcher named Schindler in the late 70s. And he worked in the experimental lakes area in Ontario, Canada, which are very isolated, very low nutrient, low algal production lakes. So what he did was, what you do is you add fertilizer to them and see what happens. So this was I wouldn't recommend this on anyone's private lake, but it's a set-aside research area for these sort of things. So what he did was he took whole lakes and added fertilizers in different amounts and measured the response, right? And he also separated basins with a curtain, basically, and then added nutrients to half the lake and saw what happened to that half versus the other. So I'm going to show you one of the more famous ones, Lake 226. Lake science people get really excited about this one. Might tell you something about who we are. But, so one basin he fertilized with carbon and nitrogen, separated it with a curtain, went from surface to the bottom, And then he fertilized another basin with carbon, nitrogen, and phosphorus. And I'll animate the slide, and I'll see if you can guess which one had the phosphorus. Yeah, the green one, right? So that was one of the more classic kind of definitive eutrophication studies that led kind of to that paradigm that phosphorus limits in low production lakes. Limit the phosphorus, you improve water quality. So these two folks, Schaefer and Oglesby, are two Cornell researchers, and in the early 70s, it was published in 1978. They discussed phosphorus inputs to the 11 Finger Lakes and 2 others. I'll just focus on the Finger Lakes for now. They were one of the first to discuss phosphorus input to lakes in terms of how usable that phosphorus was for the algae. And I'll talk more about that in a little bit. But they kind of coined the term bioavailable phosphorus, which is just what it means. It's phosphorus that biology or biota can use, phytoplankton and things like that. So what they did was they took a comprehensive look. They estimated both total phosphorus, which is TP, and bioavailable phosphorus inputs to all the Finger Lakes in the early 70s. And they did it for multiple land uses, wastewater facilities. They estimated phosphorus export per person on septic systems. They built groundwater, atmospheric deposition. They did this for all the lakes. And they also, which was interesting, they assessed a methodology to determine phosphorus loading in terms of phytoplankton usefulness, both in terms of how available it was and where it went, in terms of did it load to an area of the lake that was useful for algae, right? If that BAP input goes to the bottom, algae can't use it. So they looked at it in terms of lake morphology, in terms of lake surface area and upper mixed depth. And that's that little BAP subscript L, the bioavailable phosphorus load. And so they did this for all the lakes, and then they compared the inputs with lake TP concentration, chlorophyll A or algal concentration, and clarity for all the 11 lakes. And what they found was the bioavailable phosphorus load was highly correlated to in-lake total phosphorus concentrations and algal production. And that the higher the algal production, the lower the clarity. And that tends to make sense, but that was the first quantification of the importance of the bioavailable phosphorus term loading to these lakes in terms of kind of the magnitude of the inputs and the size and shape of the lake. Right, so the bioavailable phosphorus load was controlling trophic state, and it explained the differences in the water quality between the lakes. And that was 40 years ago. Okay, so forms of phosphorus. Now we're going to get back into the types of phosphorus and talk about bioavailability. So total phosphorus, which I'm sure a lot of you folks have heard, total phosphorus is the total amount of elemental phosphorus in a sample. So it's not really, it's kind of the sum of all the different types of phosphorus into one useful measure, right? It's the most common measurement, and that's because it's easy to sample, and it's less prone to analytical error or sampling error than some of the more kind of exotic forms. So that's kind of the standard. That's what everything's kind of been based on in terms of inputs and trophic state of different lakes. But phosphorus really, total phosphorus isn't really a thing. It's kind of the pool of all of its forms, right? So there is dissolved forms of phosphorus, DP, particulate forms of phosphorus, which is usually calculated by difference, TP minus TDP. You can break the dissolved forms down even farther into soluble reactive phosphorus or soluble unreactive phosphorus. And the particulate phosphorus can be broken down into particulate organic phosphorus and particulate inorganic phosphorus. And I think this also may say something about me, the fact that I find this topic interesting. I'm trying to make it fun so that I don't lose it. But, it'll come back full circle, I promise. So phosphorus is made up of lots of different types of different phosphorus. So the phosphorus bioavailability concept is that phosphorus exists in multiple forms, right? and that those forms differ in their ability to support primary production, or algal growth. And that those forms that are usable by algae for growth and metabolism, we call those bioavailable. Okay. So here are the 4 basic forms. I'm not expecting anyone to memorize these. But you have your soluble reactive phosphorus at the top and your particulate inorganic phosphorus at the bottom. And bioavailability, you know, we try to group things into nice categories to make sense of it. But bioavailability is actually, it's a spectrum. So the soluble reactive phosphorus, also we call it phosphate, basically is highly and instantly available to algae and phytoplankton. So it's the stuff kind of in fertilizer, right? It dissolves, you throw it in a lake, and all of a sudden the algae can take it up immediately. Soluble unreactive is also a dissolved form. It's mostly usable by algae, but not completely. There are forms that it can use and types that it can't. The particulate organic carbon would be things like organic solids that may be partially usable by algae after some sort of enzymatic process, or after the phosphorus kind of leaks off of it once it's in the lake. But not much of it is used, you know, by the algae directly. And this last one, particulate organic P, is poorly available. It's the stuff in mud, on sediment, on clay particles. It doesn't get released from the sediment when it's in the lake, and so it's not available for algae use. And so that's my, I meant to click a minute ago, that's my happy algae with the SRP and my disappointed algae on the PIP. And just as a quick aside, so the top face is what my kids make when I say we're having pizza, and the bottom one is what we're having meatloaf. So that's, you can think of that. And so, yeah, so the SRP, the stuff that's kind of instantly available is things like fertilizers, for example. That's what the algae can use. The particulate inorganic P are things like sediment, wash off, landslide, mud, stuff that is obviously not good for anybody. It makes aesthetic problems, but a lot of that stuff can't actually grow algae. And there are other reasons why it can't either. If you think about it, when a turbid plume enters a lake, it's along the surface and it looks unsightly, a lot of that stuff settles out faster than the algae can use it anyways. Okay, and so how do we know the different forms have different availabilities? Well, we can measure it. And one of the ways to do it is with an algal assay. So you take a phosphorus solution and you put some algae in it that are hungry for phosphorus, and then you measure their uptake over time and see how they either take up soluble phosphorus or particulate phosphorus, and you measure the changes. And it's kind of a preferred method because it's a direct measure of algal response to the different types. The problem with it is that it's labor intensive, both in terms of collecting the samples, filtering the samples, running the algal assays that's not done everywhere, specialty labs, and then you have to run basically the different phosphorus tests over and over weeks, right, to measure its uptake, which gets very, very expensive. But it is good because you're actually measuring what the algae do with the phosphorus you added. And this is kind of a complicated graph, but so in terms of measuring the availability of the soluble types of phosphorus, what you do is you start out with an initial concentration, right, of the unreactive soluble and then the reactive soluble. And you introduce it to a culture of phosphorus-starved algae. And then you measure the SRP and the SUP over time to see how quickly it's being taken up. And you can see here the SRP basically is all used up after 1 or 2 days. So that's 100% available because it all got used up. It's gone. There's no more SRP left. They used it up. The SUP kind of takes a little bit longer, and you can see that there's more of it left over, so it's not completely available. Does that make sense? Yeah. And so, yeah, so the amount remaining versus the initial is kind of the fraction of it bioavailable. And in this sample, the SUP was 63% available and the SRP was 100% for all using it. The particulate phase, oh, and the folks in the parentheses are the folks who kind of invented the process. That's the reference where you can go to. This isn't me. This is, smarter folks than me have figured this out a long time ago. And the particulate phase uses this neat little dual culture apparatus. So what you do is, in the dark is your sample. On the light end is your algal community, and it's separated by a membrane. So you add your phosphorus sample into the dark. The stuff that's going to come off the particulates comes off, diffuses across the membrane, gets taken up by the algae. And you measure that over time. And one thing you probably should notice, so basically this whole test is done in 12 days to reach steady state. But what that means is you're paying a graduate student to run phosphorus samples on that one sample for 12 straight days. So it's a lot of work. The phosphorus, the particulate phase, is done over 21 days and sometimes a month. So that takes some poor grad student someplace every day babysitting these algae and measuring phosphorus uptake. But one thing you can see is that it's kind of reversed because you're kind of harvesting the particulate part, but only about 25% of the particulate phosphorus was usable by algae. And this is a tried and true method. It's been done in the Great Lakes and in a lot of places in New York. The dots down by the Hudson are the New York City reservoir systems. The circle dot is the Cayuga Lake system. But the gentleman at Michigan Tech University, Marty Auer, who does these, he's done them all over the Great Lakes, in the lakes, in streams, wastewater treatment plants, to kind of get a feel for the different availabilities of these types of phosphorus. This graph, this is just to demonstrate how, I don't know if I could have made that more tricky to say, variability and bioavailability. That's for the soluble phase stuff. So I don't know if you can pick out the red and the black bars, but the red bars are from the Cayuga Lake study, which I'll talk about a bit more. And the black bars are from other sites in the Great Lakes region. But what you can see is that for the soluble phase, the SUP, it ranges from almost completely available at this end, so where it's all usable by algae, down to the other end where none of it was, from these different locations. So it's a broad spectrum, even within this phase, of what the algae can actually use of this stuff. But on the whole, most of those fraction bioavailables or percents are higher than 40% for this phase. The particulate P, on the other hand, there's a range from 0 on the far right to more than 80% here. But most of the particulate P stuff is not very usable by algae. Yeah, so the other way to measure availability is through chemical extractions. I'm not going to go through all of these, but basically through a series of chemical reactions, you get it down to 8 or 6 different categories based on how easily does the phosphorus liberate from the different particles or different solutions. This is from a paper that I'll talk about a bit from the Great Lakes. But an advantage this has is it's very cost effective. It's just basically one sample you're running in a lab. There's less labor, analysis time, but it isn't a true measure of algal response. And it can be limited analytically when you get very low numbers for some of these fractions because they're based on subtractions, and you can get negative numbers. Okay, so now I'm going to talk about how we applied this concept to the Cayuga Lake system. I'm going to talk about the tributaries, and so full disclosure, this was it's not a DEC project. This was a project done by the Upstate Freshwater Institute in Syracuse, who did the implementation of the collection. Michigan Tech University in Houghton, Michigan ran the algal assays, and this was for the Cayuga Lake monitoring program in 2013. And a lot of what I'm going to show you here summarizes the work that went into the development of this paper. listed here. So, like all the Finger Lakes, phosphorus is important to Cayuga Lake. Cayuga Lake is phosphorus limited. The nitrogen to phosphorus ratio is greater than 100 to 1. So there is excess nitrogen compared to phosphorus. So the same sort of thing holds, right? So if you control the supply of phosphorus to Cayuga Lake, you can control the algal growth. And this has usually been based on total phosphorus and the development of total phosphorus loading rates, which that just means total phosphorus inputs in terms of mass over units of time. And so a reduction in the total phosphorus input is or has been the primary management approach for improving or protecting water quality. And the goals for this project were to do that, but to also estimate the bioavailable phosphorus inputs to the lake and see what information we could gather from comparing the two sets of numbers. And again, the bioavailable forms are the forms that grow algae. And so this study was important because it quantified both TP, total phosphorus, and the parts that are just growing the algae, just the forms that are growing algae. And it was determined from inputs from multiple streams, multiple watersheds, and point sources. I'm only going to talk about streams today and not get into the point sources so much. And the information collected as part of this project was used in the development of a lake water quality model, which is going to be used for Cayuga Lake. And the results were discussed for management considerations in terms of both total and bioavailable phosphorus. So this is a map of Cayuga Lake, some of the important streams, some of the important inputs. The shelf area is highlighted here and kind of the inset. But it was an intensive monitoring program over April through October in 2013. The mouths of 5 major streams were monitored for at base flow and at high flow, which is important to kind of characterize nutrient levels at different flow regimes. The primary focus was for phosphorus, so particulate phosphorus, that SRP, that soluble reactive phosphorus, SUP, that unreactive dissolved form. And for this project, more than 200 phosphorus samples were collected from these streams. And it was one of the most comprehensive BAP studies in a single lake watershed system in New York. And so we also collected, I say we, I was on the team that did some of the work, so collected 3 samples for bioavailability assays from each of the streams, for both the soluble forms and the particulate forms. Okay, and so how did that work? So there's 4 basic steps. There's collecting the sample. One of the issues with these types of assays is it requires large volumes of water. So each sample required about 40 to 80 liters of water being collected. And it had to be collected with stream water with lots of particles because there has to be something for the algae to either to use, in the assay. And so that's number one. You'll notice that I am taking the picture in this picture and not the young man collecting the sample, which he reminded me of as he was carrying 80 liters of water up the bank. Then you take that collected water and then you filter it through standard membrane filters, which requires hours and hours of filtering. As you can imagine, that very turbid water clogs those filters very quickly. So you pass the first 5 liters through in a heartbeat, and then it could be an hour for the next 20, and then 2 hours for the next 40, right? So, but the point is to kind of, they term it building the cake. So the cake is that brown stuff on the filter on number 3. So we're trying to get as much of the particulate matter on that filter as possible. And then you send the filter with the cake and the stuff that passed through the filter off for the algal assays, which we talked about a little earlier. And I will just, folks that do stream monitoring and storm event sampling, it usually rains 1 or 3 times in the dark, on the weekend, or before a public holiday. So in this picture of Fall Creek was July 2nd, and I got the last of these samples filtered just a couple hours before fireworks on the 4th. So it's a lot of work. All right. So yeah, so that was done. So that was done for all of the different streams 3 times. So basically 15 samples altogether for these bioavailable assays. And now I'm going to talk about some results. And these are just some standard results. So this is a stack plot of stream flow on the top panel over time, right, in 2013. So you can see variable changes in flow over time. The middle panel is particulate P. You can see that the concentrations are variable, very dependent on flow, right? The particulate phosphorus peak when the stream flow is up, right, which makes sense. The dissolved forms, the SUP component was mostly less than 20 parts per billion or micrograms per liter, not extremely variable. And the SRP component, the one on the bottom here, D, was not as variable as the particulate P component, but still fairly irregular and fairly highly influenced by stream flow. And one thing you want to keep in mind is just the axes, the scale changes, right? So these are 0 to 60, and the particulate P is 0 to 600. So we're talking about orders of magnitude higher levels of particulate phosphorus. This is for Fall Creek versus the dissolved forms. That's a different trip, but that's what one of the trips to Cayuga Lake looks like at high flood. Can you see the turbidity? It's kind of hard to tell. You know, after a rain event, very brown, high water, very turbid. Okay, so Fall Creek phosphorus concentrations kind of broken down into its parts. So at low flow, the average Fall Creek TP concentration was 27 parts per billion. And you can see it kind of got broken up into its 3 components: particulate P, SRP, and SUP. So the particulate P was 61% of the TP at low flow, and the dissolved forms, rounded out at 16 and 23% for the SRP and the unreactive P. At high flow, and I just did a quick grab of flow above the long-term average for the site, you can see how much higher the total phosphorus concentration is. It's 128 parts per billion. And it was dominated by the particulate component. Right? 86% of the TP at high flow was as particulate P. And the dissolved forms, the concentrations got bigger compared to low flow, but in terms of relative contributions to total, the dissolved forms were much smaller than the particulate at high flow. So then I took all of those concentration and flow data and developed loading inputs in terms of mass into the lake. For Fall Creek, that's what this is here. This axis is mass in pounds per year coming into the lake from this one creek, okay, for each of the years, 1998 through 2013. The average load, and this is of phosphorus now, total phosphorus, was about 45,000 pounds per year for this one creek. And one thing you can see is there's a lot of variation year to year. Some years it's high, some years it's low. 2012 was the lowest year. It had about 17,000 pounds delivered by this one creek. And 2005 and 2011 had more than 80,000 pounds of phosphorus delivered. And this is just due to differences in precipitation and hydrology and stream flow. So a high runoff year like the one we had this year, the numbers will be higher. A drier year with less runoff, they'll be lower. And you can see from this graph, so this is the same graph, except for 1 year in particular, 2005, from January through December, total phosphorus load in pounds per day. The blue line is the total phosphorus load. The orange line is the stream flow. So you can see how the stream flow drives the phosphorus loading in this creek. When the flow is up, the load is up. And so I've kind of drawn that box around those dates. We're going to take a closer look at those dates. So that represents March 27th through April 11th, which is 16 days. And during that time, 52,000 pounds of phosphorus was delivered by Fall Creek, that's one creek, to the lake. That's 26 tons of phosphorus, not sediment, just phosphorus. It was 64% of the annual load in 16 days for that 1 year. That was snow melt and inches of rain just caused the whole watershed to just, export sediment and phosphorus into the lake. And that 16 days was more than the annual totals in 10 of the 16 years. So it kind of highlights the challenges of watershed management, right? I mean, if you're losing that much sediment and phosphorus in just a short period of time, it's hard to manage.
Tony,
Yes, sir.
you're extrapolating your sampling data and then utilizing, this is modeled, correct? This is not actual samples, this is a
Correct.
model.
It's daily output from a loading calculation software.
Okay.
Yes,
and
thank
it's
you.
based, yeah, no, and it's based off and calibrated to a certain extent off of the actual sampling you guys did.
Yes.
And then now you're running the model based on that data.
Yes.
Is that- Okay, I just want.
Thank you, yes.
No,
I should have taken the time,
No.
but yes. Yeah, so you take all of your concentration data
Right.
with
Yep.
paired flow data,
Right.
and you can
Okay.
develop relationships to predict the phosphorus mass per ton.
Okay.
Sorry, I should have.
No,
done
it's,
more.
yeah, I just want to make sure.
So that's a dump truck load of phosphorus, just elemental phosphorus, not sediment, right? That's a lot. And I did a quick back of the envelope of sediment, and that 16-day period delivered close to 3,000 dump truck loads from the one creek in 16 days. So that's the challenge, is how do you prevent that loss into your lakes? And that's what Cayuga Lake looked like immediately after that runoff event. And that's courtesy of Bill Heck. I'm not sure, I mean, he takes beautiful drone and aerial photography. So that's the marina at the south end, right? That's actually Fall Creek here, and this is the Cayuga Inlet. And that's from April 2005. And those numbers are just from Fall Creek. The inlet was actually higher. But I have the Fall Creek numbers on hand, so. Yeah, it's an amazing amount of stuff can get moved in one event. So over that 16-year period, this is Fall Creek again, only Fall Creek. About 720,000 pounds of phosphorus was delivered. April 3rd, 2005 was the highest day. It delivered about 29,000 pounds. The highest 100 flow days delivered 45% of that 16-year total. Pretty impressive. And I know, I've seen this graph a lot, I know Kevin likes this one, but it says heavy precipitation and increased stream flow are going to become more the norm. It just creates a heck of a challenge to kind of keep nutrients and sediment out of our lakes. Yeah.
Can your model identify the proportion that's coming from the city of Ithaca?
Well, it did. I'm not going to talk about it here. But we looked at all the point sources, all of the watershed sources. For obvious reasons, I'm going to talk about watershed sources today. But it did. Yeah. Okay. So this is watershed loading, April through October 2013, of just total phosphorus. So the total amount of mass that came in, this is the whole lake, not just Fall Creek. Again, I have this graph handy, so here it is. It was a little under 120,000 pounds of total phosphorus. 84% of it was particulate, right? And then the SRP was 10%, and the SUP was 6%. And that's from all the different creeks, none of the point sources. All right, so I spent some time talking about total. Now we'll get into some of the bioavailability issues. So as we talked about the particulate phase, so that dual chamber, we did the assays on all the different stream sources, and these are the results from 5 different streams. Right? So it was variable between the streams, but the particulate stuff just isn't very usable by the phosphorus. I mean, the highest stream, 21% of the particulates were able to be used by the algae. At the low end, it was around 6. So the particulate stuff that comes in just isn't used by algae. The soluble phase, on the other hand, is much more available for algal use than the particulate P. Right? So for Watershed 1, particulate P, 9% of it could be used, but for this form, 77% of it could be used. And the SRP phase, the phosphate, was 100% available no matter where it was collected. So you can see differences between particulate and soluble and in between watershed sources. And so this is what the loading looks like. So this is back to that pounds per year graph with TP as the blue bar. So that's the total load, the total phosphorus that came in 1998, and that's the bioavailable load. So you can see how much less available phosphorus comes in. It's part of the total, but it's not usable by the algae, so it's much, much smaller bar. And so that click, the total phosphorus stayed the same. The circles are proportional. The total bioavailable phosphorus input was only 31,000 pounds compared to 120,000 of total. So it was only 26% of the total phosphorus that came in could be used by algae. The rest of it is just attached to sediment particles and settles out of the water column, and the algae don't use it. But you can see that 44% of it was particulate versus 84% in total. And the dissolved forms were much more important in terms of the stuff that the algae could use, which highlights the importance of monitoring, when you're doing water quality modeling, monitoring all the different types of phases of phosphorus. And so the percent bioavailable compared to total was variable between years. 2005 was a wet year. The total phosphorus load was around 82,000 pounds. The bioavailable load was 14,000 pounds, or 17%. On a drier year, in 2012, for example, 28% of what came in was bioavailable. And that's due to the dominance of particulate P during wet years and the fact that a lot of that particulate P can't be used by the algae. So the differences between the two is mostly due to the low availability of the particulate stuff, the sediment stuff that comes in. All right, so I know you're thinking it, so I'll just put it up there. Hopefully it's not easier. So in terms, other than theoretically and running lake models, why would anyone care about this? Well, it has important information for watershed managers and lake managers. So here's a nice picture of the Cayuga Lake watershed. Okay, each of our 5 streams again, their respective areas. And this is the annual average TP load in pounds per year for each of these streams. Okay? Stream 4, over the 16-year period, had the highest average TP load. And on an area basis, so you take that pounds per year and divide it by its watershed size, and you get 3 pounds per acre per year from that stream, which was the highest by far in terms of total phosphorus to the lake, which ranks it as the highest. So you would target that one for best management practices, or you would target it for erosion control measures. You would use that information somehow. And that's the total. So now I'm going to click, and I'm going to put it on the bioavailable loading per unit area. And what you can see is where stream number 4 was the highest rank in total phosphorus, stream number 5 is the highest in the bioavailable phosphorus, the stuff that's growing the algae. Okay, so in terms of pounds of bioavailable phosphorus per year, number 5 is twice higher than stream number 4, twice as high, and almost 4 times higher than the other ones. So it has real value. Not that you'd ever discount the erosion component. You'd never let that go. You'd never say, I'll just let that ditch go, or that field is sloughing off, so it's fine. It's not growing algae, right? But it's important to know both, because if, for example, you had 2 similar streams and one of them had much higher bioavailable concentrations and similar TP concentrations, well, then you could make a decision on reducing the stuff that's actually growing the algae. Okay, so we'll get into some other case studies. And that's my reminder. I wasn't forced to write this, but I thought it was a good idea. This isn't a DEC study either. What I'm going to do now is talk about some research from the Great Lakes. But I find it interesting. Hopefully you will too. But I just wanted to make sure that, you know, I'm not promoting it. I'm just kind of sharing the information. Okay. So we'll talk about Lake Erie a little bit. In the 1960s, Lake Erie had a lot of water quality problems. It was very eutrophic, okay, which led to legislation and International Joint Commission efforts to reduce phosphorus loading to help improve water quality in Lake Erie. And initially it had worked. They were labeling it a success story until about the mid to late 1990s, when eutrophic conditions resumed in the western basin of Lake Erie, including the development of these pretty nasty algal blooms, of which I'm sure a lot of you have seen the aerial photos, right? And this is one from the Toledo Blade. We've all heard about the drinking water concerns in Toledo and things like that. So these researchers, Baker and others, in 2014, they evaluated a long-term total phosphorus and bioavailable phosphorus data set from Heidelberg University. They looked at 3 of the largest rivers to the western basin of Lake Erie, and they assessed trends in total P and bioavailable P in these rivers as inputs to Lake Erie, and they had data going back, I think I said, 30 years. So from the 70s and 80s, they had data. And what they found was there were large decreases in non-point total phosphorus into Lake Erie, mostly due to reducing the particulate P being transported. But they saw increases in the bioavailable phosphorus export for those same rivers. So the total numbers are down, particulates are down, but the bioavailable stuff, the stuff that's actually growing the algae, is actually higher now than it was in the 80s. Another group, and some of them are the same authors, but Kane and others, they looked at the lake itself. And what they saw in the mid-1990s, increases in phytoplankton biomass in the western part of Lake Erie. And they found significant correlations or significant relationships between the total phytoplankton and cyanobacterial biomass and the bioavailable phosphorus inputs to the basin. So they correlated the two, the inputs of the stuff that actually grows algae and the algae itself in the lake. And so they concluded that the increases in soluble P from the watershed were likely, I highlighted it, contributing to the increases in algal growth and harmful algal blooms that they were seeing in Lake Erie. And I have these citations. I can make them available to people if they're interested in reading further. And so the big question is why? Why, if you're cutting the total P, why is the soluble P coming up, the bioavailable P? Well, they speculated, of course, the scientists do. No one wants to put a definitive answer on anything. But so they speculated that erosion control measures, which are very good at reducing particulate P export, were actually making the soluble P export worse. And they gave literature, and I can provide that to you, of specific control measures that can actually make the SRP, or the bioavailable P phosphate export from watersheds higher. And they also noted that stream flow is increasing in the region due to hydrology modification and climate change. And again, that's their take on what they saw in their data. Because one thing it wasn't a completely comprehensive paper, right? They just looked at nutrient export and the lake response. They didn't talk about climate change, for example. They didn't talk about the role of invasive mussels and nutrient recycling and selective grazing by some of these things. And they obviously didn't look at the array of emerging contaminants that may be responsible either. Okay. I'm almost on time. So, are everyone still awake? Okay, good. So, it just makes for another hard decision, right? I mean, the stuff that grows algae, that's good at growing algae, fertilizer, septic tanks, sewage, runoff from farm fields, is just one more layer to have to consider versus just the stuff that is important, like erosion, landslides, stuff that you'd never let slide. You have to take care of those problems. But that stuff on the right with the unhappy algae, while important aesthetically and to the value of people's land, probably isn't growing a lot of algae in the lake. And so best management practices, watershed planning, construction of stormwater programs, nutrient reduction strategies from farm fields and people's lawns and things like that are all very important. And I took a quote from the Baker paper. They concluded with phosphorus load reduction programs should address both, they called it DRP, or the bioavailable phosphorus, and particulate P, and take into account the lower availability of the particulate forms. And I think I'll just close with just how hard that is given the climatic times we live in terms of more frequent rains, higher intensity rains, that just whatever sort of management activities take place, they have to plan for more frequent, intense rain events. And that's it. And I'm 3 minutes over. So. Yeah.
You talked almost exclusively about external sources.
Yes.
Can you comment anything about internal sources from an oxy at the bottom?
Yeah, it varies.
What kind of phosphorus is that?
It's
all soluble reactive?
It's all soluble reactive. Yeah. So in shallow lakes that are more productive, if the lower waters lose oxygen in the summer through normal respiration processes by bacteria, without oxygen in the water, the phosphorus can be released to the sediments into the water column, and it's all dissolved forms. It's all SRP. And then when the lake mixes, that stuff's available for the algae. And the same holds true for the kind of the common thinking with the zebra mussels is or quagga mussels, they'll take in particulate forms of sediment, organic matter, through their normal processing. They excrete highly usable dissolved forms.
Yeah, well, in Honeymoon Lake, in the summertime, the predominant source of phosphorus is from the internal.
Yep. Yeah, it's like that for a lot of more productive shallow lakes. And the zebra mussel and quagga mussel component is interesting, and that's one thing they didn't talk about in either of the papers that I talked about. But there's a lot of research coming out of the Great Lakes at just how important they can be in terms of recycling unusable forms to usable forms and making that available to the algae. And that's in real time because they're in the surface waters where the algae are.
I have 2 questions, Tom. Just 2.
I hope to have 2 answers.
Could you repeat the
First
question?
question.
Oh, yes.
First question is, you've got a lot of oligotrophic lakes here, Canandaigua, Skaneateles, Keuka. And in the last year to 3 years, they have started to show blue-green algae blooms in the late summer period.
Yes.
But the total phosphorus, which measures all types of phosphorus, is single digits, 6, 7, 8 micrograms per liter. The EPA criteria for an impaired lake is like 20 micrograms per liter.
Yes.
So they're like 1/3 of the EPA criteria, yet they're having blue-green algae blooms.
Yes.
So that was just a lead up is, have you seen any studies as to how low you have to get
Yeah.
the phosphorus?
Yes, I'll, do I have to repeat? Was that audible? Should I repeat? Okay.
Yes, repeat.
Yes, repeat.
Yes, repeat. Okay, so what Cherry said was there's a lot of very low phosphorus lakes like Canandaigua, Skaneateles, Keuka, other lakes, Lakes Placid, that are very low phosphorus, but still having algal bloom problems.
Yes.
And what do I know about that, or what was the research kind of showing? Unfortunately, nothing conclusive. There's a lot of pieces. One thing is having a handle on how much bioavailable phosphorus is coming in, I think is going to be important. Because that stuff, if it's kind of coming in and getting hung up near shore before it makes it out to the open water, because most of the total phosphorus numbers are collected at a representative open water location, not right near the shore, right? So that's one thing. The other thing is that there is some research for some folks, I'm trying to remember, Rakau is one, Sarnelli, they are investigating the relationship between blue-green algal response and dry-scented mussels. And there's some indication that the blue-green response in the presence of mussels can be worse in low phosphorus lakes. And I can provide those references to you.
Oh, that would be very interesting.
It's that, I mean, it's, scientists, they don't agree on much, but I can provide those.
Yeah,
Yeah.
so I'll stay away from Canandaigua Lake because you got the experts in the room. I'll pick on Scanny Atlas and Cuca.
Yeah,
They showed up with blue-green algae blooms first time in
right.
ever this summer. Any thoughts about why?
None other than the two I just gave you.
Same.
Yeah, I mean,
But you have no data.
yeah,
to-
well, one thing that'll be nice this year, and I'm behind in terms of, I think I saw Bruce leave, I need to grab him, but he has a nice long-term data set for Canandaigua. There are other similar, kind of more sporadic data for a lot of the other lakes that one of the charges is to kind of accumulate all that and see if we can see any sort of trends in water quality that may be indicative to this occurrence on these lower phosphorus lakes. The Citizen Statewide Lake Assessment Program the DEC sponsors was on all 11 Finger Lakes this year, bi-weekly, at one or two locations.
Yeah.
And we looked at not just nutrients, but algal community composition and toxin indicators. So once we get those data, that'll be very insightful to see if anything kind of shakes out in terms of the lead up to kind of the late summer blooms on Skinny Atlas and Cuba. One thing that was kind of common to the region, and I'm sure all of you know, there was that stretch kind of in mid-September where it was hot and not a breath of wind, and that's exactly the types of conditions that these things like.
Absolutely.
Yes.
Has anybody done any studies on the ratio of total algae to blue-green algae at different times, like when there's an algae bloom?
Yeah.
Blue-green algae bloom, is the ratio different than, for instance, in June, when they don't have?
So the question was differences in the algal community between kind of the acceptable algae and the blue-greens. Yeah, that's been done quite a bit. In a few of the lakes that I've looked at, normally, even in normal conditions, the cyanobacteria, the blue-green algae, are in the water column all year. Normally a very, very small component. It's actually a natural kind of succession in the algae to get a blue-green kind of dominance, or not dominance, but kind of higher numbers into the late summer and fall. That's not uncommon. Kind of these widespread prolific blooms where you see them from drone shots on Skinny Atlas, for example. That's new to Skinny Atlas, but a lot of folks have done that. One thing that will be, that sort of data was collected all season, so we'll have those data to look at this winter.
How does pH affect the amount of available phosphorus?
Oh, good question. I'm not going to have a good answer for you. I'm going to have to dig back into my chemistry stuff. And,
We've had this period of so-called acid rain, which is now declining somewhat in the northeast.
yeah, I
Just wondered if there's a correlation there.
Yeah, it's a good question. The Finger Lakes as a whole are fairly high pH lakes, fairly resilient to the acid rain problem, so I don't have a good answer for you. I don't think it would be pH driven, but I'd have to look to be checking. Yes?
I have three questions. One, how is it that you determine SUP, and then how long does the SRP stay around? I mean, I know phosphorus as element can be created or stored, but I mean, as far as
Yeah.
available in the lake, and then what, like some of those loadings during that Fall Creek study, there was a big loading of that particular phosphorus. What's the significance of that?
Okay, so two questions. One was, I'm sorry, which
Three.
one?
The first one was.
How do you calculate the different forms?
Well, how do you determine SUP? I know how you got, I used to be working in the lab, I know how to analyze for SRP.
Yeah, I kind of went quickly over that. So you analyze for SUP by taking the total dissolved measurement and subtracting off the SRP. So it's by difference.
Okay.
So it's just a
So
analytical
you go through
calculation.
the digestion for total phosphorus on a dissolved sample, and then you analyze for SRP, and then you subtract it.
Correct, exactly,
Okay.
exactly. And how long does it hang around in the water column after it's delivered?
Yeah.
The SRP it can be used almost instantly as it comes into a lake. SRPs in the tributaries are normally much higher than the lake themselves, than in lakes, because once it hits the lake, the algae are constantly taking it up. Now it recycles, when an algae cell dies and lyses, it'll become available, bacteria will mobilize it, but then it's almost immediately taken back up. The SUP is a little bit longer. That time, if you remember the two curves with the,
Right.
it's taken up on the order of the day, very quickly.
And then the PP, the particulate phosphorus, what's the significance of that?
Yeah, that was me just showing kind of the magnitude in terms of a big particulate phosphorus load, which is visually striking and important because it's erosion and sediment loss. But as nasty as that looked, that's not the stuff that's growing algae.
But that will eventually settle, right, in the bottom of the lake?
Correct.
And then, under anoxic conditions, it could be digested
It
by
could.
bacteria and filtered back into the water.
It could, and a lake like Cayuga doesn't go anoxic, so
Right.
it wouldn't be as important in Cayuga. Now, some of that stuff could be digested by a zebra mussel, for example.
Right.
That particulate stuff and kind of spit back out as it dissolved. But for the most part, in kind of the deep open water lakes, that stuff will just settle out of the photic zone where the light is, below the epilimnium, and not be available for algae.
Right. Okay.
It was more to show the nasty photo of the sediment plume
You know, it's-
in relation to its availability.
Okay.
Tony, is it available for macrophytes though?
The particulate P would be available for
Right,
that.
so that's, I think, from a watershed management perspective, that's the other, especially for a lot of the folks along the nearshore area, it's when we get these huge sediment plumes coming in from these small and large tributaries, it has that impact also, or it lays this beautiful garden of
Yes.
sediment and phosphorus, and when you get all the vegetative growth, which is another part of the equation of what we look at. So,
Yeah,
yeah.
the bioavailability part of this study was just algal growth. Not-
Right, yes.
Not macrophyte, exactly. Yep, but, macrophytes, like true plants, get their nutrients from the sediment, from
Right.
roots. So it definitely replenishes the sediment phosphorus pool,
Yep.
for sure, the particulate stuff. Yes?
Is the relatively high N-to-P ratio relative to marine environments, which you talked about much earlier, a natural phenomenon, big limnology you're speaking of, or is that also influenced by human activity in the watershed? Does the elevated nitrogen come from nitrate sources?
Yeah,
Other than
so,
that.
yeah, that 7-to-1 is the amount of nutrients in the algae themselves, not the ocean water.
Okay.
So that's what they need. They need 1 part phosphorus for every 7 parts nitrogen. That's what the algae need. And the water in the Finger Lakes is overloaded with nitrogen in excess, so that's how you know that the phosphorus is limiting. I don't have a good feel for what N-to-P is in ocean water. I don't have a good feel for it.
And do you have a good idea about the N-to-P ratio in Canandaigua right now, as opposed to the 20-year?
I will know as soon as the sea slap data comes out. It's on the to-do list for sure. Yeah. I mean, given looking at the results that Bruce has published in the management plan, et cetera, where the total phosphorus numbers haven't changed a lot over the years, I expect it to show a similar story. The exact numbers might not be the same, but I don't expect it to be anywhere near limiting for nitrogen, for example.
Yeah, we're typically around 5-6 micrograms per liter for total phosphorus and roughly, say, 300 micrograms for total nitrogen. So it's about a 60 ratio from that perspective.
Yeah. Well in excess of what the algae need for nitrogen.
Mm-hmm.
Excuse me.
Any other questions for Tony?
I had one more. And is there any source, recognizable or hypothesized source for the SRPs?
Oh, in terms of
Like
what's
what-
coming?
Right.
Yeah, I mean, it comes from the same basic
It's
sources.
not, right, it's not the stormwater runoff, and it's not the,
Yeah, well, it's in the soil. It can be generated in the soil by microbes. It can be septic tank effluent, manure runoff, normal processes in the soil, fertilizers.
So I'll ask one last question, and maybe Kevin, you know, of our wastewater treatment plants, I know Canandaigua does not, but do any discharge into Canandaigua Lake?
So
Or
you have
into
Rushville's
tributaries?
plant discharges into the West River, and there's, you know, about 6-7 miles of stream and a wetland that can provide a nice biological process there. And then you have Bristol Harbor that does some tertiary treatment, and then they reuse a lot of their wastewater back onto the golf course, so you get some nice filtration from that perspective also.
And do they have phosphorus stripping as part of their, either of those treatment plants?
So, no, but I think, you know, with the way they work their plants, it reduces that impact. And it's a very small, like the Bristol Harbor one, you know, it's not producing a lot per se. We have sampled baseline conditions, Seneca Point and upstream in terms of what's coming through there. You do see a little tiny, but nothing of a magnitude, I would say.
Yeah,
Is there ever any discussion about, like, with the CAFOs having some sort of phosphorus scrubbing in their?
Oh, I mean, they are permitted facilities, right? So they get inspected. I don't know too much about the specifics of treatment. I know that they, you know, as part of their permit requirement, they are inspected and they have to maintain no runoff given certain size runoff events and things like that. But specifics of CAFOs, I don't know.
Is there any discussion within the DEC about with what is happening with the climate of maybe changing some of the?
Yeah, the design of, you know, we need to design for 1 in 500 year storms because they happen 3 times a year now. Yeah, no, that's, yes. Yeah, no, it's, the DEC is, I don't have it tip my fingers, but they have a whole climate aid resiliency program. I could forward that on to Wade or Lindsay and they could pass it over. No, it's important.
Thanks everyone for attending. Last viewpoint session of the year. I know Rob and his committee have some great ideas about the next quarter and the next year's presentation, so keep your eyes out for that. If you are a member, you've recently received your membership renewal, and if you've already sent it in, thank you very much. If you haven't, think about it. If you're not a member, please consider joining our organization so we can keep up events like this and the other things we're doing. And how about one last appreciation for Tony?